Okay, starting chapter 7, cellular respiration and fermentation. Living cells require energy from outside sources. Some animals, such as giraffes, obtain energy by eating plants, and some animals feed on other organisms that eat plants. So, giraffe. Energy flows into an ecosystem as sunlight and leaves as heat. Photosynthesis generates oxygen and organic molecules, which are used as fuel for cellular respiration. Cells use chemical energy stored in organic molecules to regenerate ATP, which powers work. So here is the cycling. This is basically the carbon cycle, or part of the carbon cycle, where you have photosynthesis that is occurring in chloroplasts, which makes organic molecules, such as glucose, and makes oxygen as a byproduct. And then those organic molecules and oxygen are used by the mitochondria to produce carbon dioxide and water, and also generating ATP by the breakdown of those organic molecules. And that ATP is used for cellular work. So you can see that it is a cycle. So then the products of cellular respiration are used as the reactants of photosynthesis. Several processes are central to cellular respiration and related pathways. The breakdown of organic molecules, such as the breakdown of glucose, is exergonic, meaning that it releases energy. Fermentation is a partial degradation of sugars that occurs without oxygen. Aerobic respiration consumes organic molecules and oxygen and yields ATP. So aerobic means in the presence of oxygen. Anaerobic respiration is similar to aerobic respiration, but consumes compounds other than oxygen. Cellular respiration includes both aerobic and anaerobic respiration, but is often used to refer to specifically aerobic respiration. Although carbohydrates, fats, and proteins are all consumed as fuel, whenever we look at the process of cellular respiration, it always starts with glucose, C6H12O6, reacting with 6 molecules of oxygen, which yields 6 molecules of carbon dioxide, 6 molecules of water, and energy in the form of ATP and heat. So what we want to look at when we look at both cellular respiration and photosynthesis is the flow of the electrons, because it is the electrons that are the most important part of both processes. The transfer of electrons during chemical reactions releases energy stored in organic molecules. This released energy is ultimately used to synthesize ATP. Chemical reactions that transfer electrons between reactions are called oxidation-reduction reactions, or redox reactions. In oxidation, a substance loses electrons. In reduction, a substance gains electrons. And of course, the reason why that is because when a substance gains electrons, it's gaining negative charges. So we say that that substance is reduced. Okay, so this is just an example of a redox reaction. Sodium is losing an electron, therefore it's becoming oxidized, and chlorine is gaining an electron, therefore it's becoming reduced because it's gaining a negative charge. The electron donor is called the reducing agent, and the electron acceptor is called the oxidizing agent. Some redox reactions do not transfer electrons, but change the electron sharing in covalent bonds. An example is the reaction between methane and oxygen. Okay, so here we have methane. You can see that the electrons are shared equally. Okay, this is a nonpolar covalent bond. And then what will happen is methane will become oxidized into carbon dioxide. So basically, you can see that the electrons for carbon dioxide are not shared equally. That's why we say that CH4 or methane has become oxidized, because we go from an equal sharing to an unequal sharing of electrons. And then oxygen will become reduced because look how much closer the electrons are to that oxygen. Redox reactions that move electrons closer to electronegative atoms like oxygen release chemical energy that can be put to work. During cellular respiration, glucose is oxidized and oxygen is reduced. Organic molecules with an abundance of hydrogen, like carbohydrates and fats, are excellent fuels. And the reason why that is when we look at the transfer of electrons, what we're looking at is we're actually looking at the hydrogen ions, or the hydrogen elements rather, because it is the hydrogens that carry an electron. So electrons move in the form of hydrogen. As hydrogen with its electron is transferred to oxygen, energy is released that can be used in ATP synthesis. Okay, so here is your general equation for cellular respiration. Glucose becomes oxidized because it's losing hydrogens. The purpose of this process is to strip hydrogens and therefore electrons from glucose. So glucose becomes oxidized because it loses electrons. Those electrons travel as hydrogen atoms. And then oxygen becomes reduced because it's picking up hydrogens and electrons with those hydrogens. And then energy gets produced as a result. In cellular respiration, glucose and other organic molecules are broken down in a series of steps. Electrons from organic compounds are usually first transferred to NAD+, a coenzyme. NAD+ is an electron acceptor. Functions as an oxidizing agent during cellular respiration. So NAD+ picks up electrons and brings them to the electron transport chain, where, as you know, most of the ATP is produced. NAD passes the electrons to the electron transport chain. Unlike an uncontrolled reaction, the electron transport chain passes electrons in a series of steps instead of one explosive reaction. Oxygen pulls electrons down the chain in an energy-yielding tumble. So remember that oxygen is very electronegative, highly electronegative. So that's why the oxygen molecules pull the electrons down the electron transport chain, because it is so electronegative. It is more electronegative than any other molecule in the electron transport chain. So that's why oxygen functions as a very good final electron acceptor. The energy yielded is used to regenerate ATP. Harvesting of energy from glucose has 3 stages. Glycolysis, which breaks down glucose into 2 molecules of pyruvate. Pyruvate oxidation in the citric acid cycle, which completes the breakdown of glucose. And oxidative phosphorylation in the electron transport chain, which accounts for most of the ATP synthesis. For the purposes of this lecture, glycolysis will be teal, pyruvate oxidation in the citric acid cycle will be salmon, and oxidative phosphorylation, electron transport, and chemiosmosis will be violet. Okay, so the first thing that happens is glycolysis, and what happens during glycolysis is glucose gets split into 2 pyruvates. A small amount of ATP is generated, and electrons are picked up by NAD+, becoming NADH. And NADH will bring those electrons to the electron transport chain. Then pyruvate enters the mitochondria. It gets converted to acetyl coenzyme A through a process called pyruvate oxidation. And then acetyl coenzyme A will enter the citric acid cycle, which will complete the breakdown of glucose and strip basically the rest of the remaining electrons from glucose. These electrons will be picked up by my electron carriers, NADH and FADH2, and they will be brought to and dropped off at the electron transport chain, where oxidative phosphorylation occurs, which involves electron transport and chemiosmosis. The process that generates most of the ATP is called oxidative phosphorylation because it is powered by redox reactions. Oxidative phosphorylation accounts for almost 90% of the ATP generated by cellular respiration. A small amount of ATP is formed in glycolysis in the citric acid cycle by substrate-level phosphorylation. For each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes about 32 ATPs. So this is substrate-level phosphorylation. We have a substrate with a phosphate attached, and that substrate will give up its phosphate and add it to adenosine diphosphate, creating ATP. And there's an enzyme that catalyzes this process. Okay, glycolysis. Sugar splitting. Glycolysis breaks glucose down into 2 molecules of pyruvate. Pyruvate is just basically glucose split in half. Glycolysis occurs in the cytoplasm and has 2 major phases: energy investment and energy payoff. Glycolysis occurs whether or not oxygen is present. Okay, so here it is, occurring in the cytoplasm. So here's energy investment. 2 ATP is required for glycolysis. However, 4 ATP get generated. So that is a net production of 2 ATP. In addition to that, when electrons get stripped, 2 NADHs get formed. So the 2 NADHs will bring electrons to the electron transport chain. You don't need to know all this, but you should be able to analyze this diagram. Like, for example, if we go back, these enzymes are written below. And then, what was the other thing? Oh, you should be able to say that here, NAD plus is picking up electrons forming 2 NADHs. So don't memorize this chart, but know how to analyze it, know what it's saying. In the presence of oxygen, pyruvate enters the mitochondria where the oxidation of glucose is completed. Before the citric acid cycle can begin, pyruvate must be converted to acetyl coenzyme A, which links glycolysis to the citric acid cycle. Okay, so we've broken down glucose in half into 2 pyruvates, and through pyruvate oxidation, that forms acetyl coenzyme A, which will enter the citric acid cycle. Okay, so pyruvate enters the mitochondria, gets converted to acetyl coenzyme A. NADH gets produced. That NADH will bring electrons to the electron transport chain. Acetyl coenzyme A enters the citric acid cycle. Per turn of the citric acid cycle, you have the generation of 3 NADHs, 1 ATP, and 1 fat H2. So fat H2, that's another electron carrier that will actually drop the electrons off at the electron transport chain at slightly a lower energy level than NADH does. Same function, however. Okay, so per turn we're generating 1 ATP, 3 NADHs, and 1 fat H2. Per glucose molecule, we have to multiply that by 2 because 2 pyruvates equal 1 glucose molecule. So per glucose molecule, you generate 2 ATP, 6 NADH, and 2 fat H2. And again, citric acid cycle occurs in the mitochondria and generates a little bit of ATP through substrate level phosphorylation. Not sure what just happened there. Okay. And then again, here is the cycle. Don't memorize this, but you should be able to analyze it. The citric acid cycle has 8 steps, each catalyzed by a specific enzyme. The acetyl group of acetyl coenzyme A joins the cycle by combining with oxaloacetate, forming citrate. The next 7 steps decompose the citrate back to oxaloacetate, making the process a cycle. The NADH and fat H2 produced by the cycle relay electrons extracted from food to the electron transport chain. Okay, following glycolysis in the citric acid cycle, NADH and fat H2 account for most of the energy extracted from food. So these are electron carriers that will bring their electrons to the electron transport chain. And those electrons will fall down the electron transport chain, generating energy, and it's an exergonic fall. They'll fall down to oxygen, which is the final electron acceptor. The electron transport chain is in the inner membrane or the cristae of the mitochondria. Most of the chain's components are proteins, which exist in multi-protein complexes. The carriers alternate reduced and oxidized states as they accept and donate electrons. Electrons drop in free energy as they go down the chain and are finally passed to oxygen, forming water. Okay, so in the mitochondria, we have oxidative phosphorylation, which generates lots of ATP. Notice that NADH drops electrons off at a higher energy level than fat H2 does. So NADH actually generates a little bit more energy than fat H2 is able to. And then the electrons fall down the electron transport chain. So a molecule picks up an electron, becomes reduced, gives it to the next one, and it becomes oxidized. This one becomes reduced until it passes the electron off to the next one, and so on and so forth. And eventually these electrons will fall all the way down to oxygen, which is the final electron acceptor, making water. Now this is an exergonic fall. As the electrons fall down the electron transport chain, that generates energy. This energy is not ATP. It's not the ATP that gets generated through oxidative phosphorylation. Instead, it drives another process which generates ATP. Electrons are transferred from NADH, or FADH2, to the electron transport chain. Electrons are passed through a number of proteins, including cytochromes, to oxygen. The electron transport chain generates no ATP directly. It breaks the large free energy drop from food to oxygen into smaller steps that release energy in manageable amounts. Okay, so how does this energy lead to the production of ATP? Electron transfer in the electron transport chain causes proteins to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. This is active transport. It's pumping hydrogen ions from the mitochondrial matrix, where there's not a lot of them, to the intermembrane space, where there is a lot of hydrogen ions. Then the hydrogen ions diffuse back through ATP synthase back into the mitochondrial matrix, generating ATP. So ATP synthase uses the exergonic flow of hydrogen ions to drive phosphorylation of ATP. This is an example of chemiosmosis, the use of energy in a hydrogen ion gradient to drive cellular work. This is just a picture of ATP synthase. The energy stored in a hydrogen ion gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis. The hydrogen ion gradient is referred to as a proton motive force, emphasizing its capacity to do work. So again, this is where most of your ATP gets generated. So here's a picture of that. Here we have NADH and FADH2 dropping electrons off at the electron transport chain, and they're falling down the electron transport chain from molecule to molecule. This is an exergonic flow, so this generates energy, and that energy is used to pump hydrogen ions from here, the mitochondrial matrix, into the intermembrane space, where there is already a high concentration of hydrogen ions. Then those hydrogen ions diffuse back down their concentration gradient through ATP synthase, making ATP. So energy flows from glucose to NADH, or FADH2, to the electron transport chain, to the proton motive force, to ATP. So here is an overview of how many ATP get made in each part of cellular respiration: 2 in glycolysis, 2 in the citric acid cycle, and 26 to 28 in oxidative phosphorylation. Most cellular respiration requires oxygen to produce ATP. Without oxygen, the electron transport chain will cease to operate. In that case, glycolysis couples with fermentation or anaerobic respiration to produce ATP. So without oxygen, you don't have that final electron acceptor. So electrons will get stuck in the electron transport chain, and you'll stop making ATP. So that's why you must switch to either fermentation or anaerobic respiration if there's no oxygen available. Anaerobic respiration uses an electron transport chain with a final electron acceptor other than oxygen, for example, sulfate. Fermentation uses substrate-level phosphorylation instead of an electron transport chain to generate ATP. Fermentation consists of glycolysis plus reactions that regenerate NAD+, which can be reused by glycolysis. 2 common types are alcohol fermentation and lactic acid fermentation. In alcohol fermentation, pyruvate is converted to ethanol in 2 steps. The first step releases carbon dioxide from pyruvate, and the second step reduces acetaldehyde to ethanol. Alcohol fermentation by yeast is used in brewing, winemaking, and baking. Okay, so the first one, glucose, undergoes glycolysis, which creates 2 pyruvates. And then what happens is carbon dioxide comes off of pyruvate, and that generates acetaldehyde. And then acetaldehyde gets converted to ethanol when NADH adds electrons to it. And then NAD+ gets regenerated so it can be reused in glycolysis. This is lactic acid fermentation, a little bit different. Glucose gets converted to pyruvate, and then pyruvate, through picking up electrons from NADH, gets converted to lactate. In lactic acid fermentation, pyruvate is reduced by NADH, forming lactate as an end product with no release of carbon dioxide. Lactic acid fermentation by some fungi and bacteria is used to make yeast and yogurt. Human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. So sure some of you have heard of lactic acid buildup in your muscles. So this would be like if you're doing an anaerobic activity like sprinting, and your cells can't get enough oxygen in time, your muscle cells will switch to lactic acid fermentation. Okay, comparing fermentation with anaerobic and aerobic respiration. All use glycolysis to oxidize glucose and harvest chemical energy of food. So it is believed that glycolysis is a very universal process that evolved early in the evolution of organisms. In all three, NAD+ is the oxidizing agent that accepts electrons during glycolysis. The processes have different final electron acceptors in organic molecules such as pyruvate or acetaldehyde in fermentation, and oxygen in cellular respiration. Cellular respiration can produce way more energy than fermentation can. So it's a much more efficient process because it is able to fully break down glucose and generate about 32 ATP, whereas fermentation only produces 2 ATP per glucose molecule. Obligate anaerobes carry out only fermentation or anaerobic respiration, and it cannot survive in the presence of oxygen, so they're poisoned by oxygen. Yeast and many bacteria are facultative anaerobes, meaning they can survive either using fermentation or cellular respiration. In a facultative anaerobe, pyruvate is a fork in the metabolic road that leads to two alternative catabolic routes. Okay, so here we have glucose getting converted to pyruvate. If oxygen is present, then pyruvate will enter the mitochondria, be converted to acetyl coenzyme A, and then it will enter the citric acid cycle. If oxygen is not present, fermentation will occur. Ancient prokaryotes are thought to have used glycolysis long before there was oxygen in the atmosphere. Very little oxygen was available in the atmosphere until about 2.7 billion years ago, so early prokaryotes likely used only glycolysis to generate ATP. Glycolysis is a very ancient process. It evolved early in the evolution of organisms. Glycolysis and the citric acid cycle are major intersections to various catabolic and anabolic pathways. Catabolic, those that break down. Anabolic, those that build. Catabolic pathways funnel electrons from many kinds of organic molecules into cellular respiration. So when we're talking cellular respiration, we're not just talking about the breakdown of glucose. Other molecules, organic molecules, are also broken down by cellular respiration. So glycolysis accepts a wide range of carbohydrates. Proteins must be digested to amino acids, and amino groups must be removed before amino acids can feed glycolysis or the citric acid cycle. Fats are digested to glycerol and fatty acids. Fatty acids are broken down by beta oxidation and yield acetyl coenzyme A. An oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. Okay, so this is just showing that not only glucose gets broken down by cellular respiration. Proteins, and other carbohydrates, and fats also get broken down, but they have to be broken down into their monomers before those monomers can enter cellular respiration. And notice that they enter at different stages. The body uses small molecules to build other substances. So for example, it uses amino acids to build proteins or polypeptides. Some of these molecules come directly from food, others can be produced during glycolysis or the citric acid cycle.